Hi, my name is Greg Owens, and I'm an assistant professor at the University of Victoria. I'm going to tell you about the non-adaptive evolution of chromosomal inversions. Chromosomal inversions have been kind of hot recently because of their potential role in adaptive evolution. A lot of theory predicts that to have local adaptation, you need to effectively link together multiple alleles, and inversions suppress recombination between orientations, effectively linking together potentially adaptive alleles. So this has been seen in Helianthus pedularis, which adapts to sand dunes in two different locations. And in each case, there's very large differences in inversion haplotype frequency when compared to sort of just general SNP allele frequency differences, suggesting an important role in inversions. We also know that inversions accumulate without any sort of adaptive purpose, that if you look at any two species and compare their genomes, they're going to differ at some inversions. So we're really interested in measuring the rate of accumulation of inversions without sort of an adaptive significance. So if we just looked at any two species, how fast do inversions accumulate and where? To do this, we aligned chromosome scale genomes for pairs of species in 30 plant genera and identified inversions based on these alignments. All this work was done by my master's student, Kaida Hirabayashi. And so what we find is that inversion accumulation doesn't really seem to be affected by sequence divergence. We might have expected there to be more inversions if there was more sequence divergence. We also didn't see more inversions if there was a larger genome, and we didn't see more inversions if there was higher TE content, all what we would have previously predicted. We also looked at sort of chromosome size under the idea that longer chromosomes are going to have less recombination per base pair and perhaps more inversions because recombination is generally bad for the fixation of inversions. But we don't see this to be true, that there is a sort of idiosyncratic patterns per genus here. We did find that inversions do tend to occur in regions of the genome with higher TEs and with fewer coding sequences. So they're typically away from genes and typically closer to TEs. So what this means is that inversion accumulation is a really complicated process, and it could be due to sort of unseen instances of natural selection, or it could be variation in the mutational process, sort of how inversions are actually occurring. And we do support with prior hypotheses about where inversions tend to occur, and we think they occur in sort of less functionally important regions. And I'd like to thank you for listening.